So they were on the video, and now they appeared here in live. Thank you very much to be here with us. Philippe Entraille, your research director, head of department Myrcène, CSO of the National Infrastructure Narratrice of CEA. Roger Legrand, your head of research center and immunology of viral infections and autoimmune disease and head of infection disease models for Innovate Therapies. IDMIT, it's a very important platform we're going to talk about here in CEA. Then Nicolas Tournier, your imaging pharmacologist from CEA. We've just heard about you. It's very interesting, what you propose, the radiolabelization. Radiolabel, it's very interesting. We'll talk about that again. Then Vincent Lebon, you're professor of medicine at Paris-Saclay University Hospital and head of department CEA. And this is for the CEA platform's representation. We already have a great view of what you did, but we're going deeper. And now to, to get the industry point of view, we have Jean Lang from Sanofi Pasteur, one of the greatest experts of vaccines, and you were involved in the race, and we're going to talk about that namely. And Magnus Fontes, your director at Institut Roche. Thank you to all of you to participate. Philippe Entraille. Let's start with you, and let's talk about Myrcène, which is an incredible platform. It's probably a unique tool in the world. From cell to large animals, you can go to any type from the smallest to the biggest.
Yes, actually, I got the idea of Myrcène when I was in Boston, so it's a strange story,
Boston.
but hi, Boston. And I realized there that when we were trying to develop a gene-based therapy in large animal models, we were lacking a tool in which a big building in which we could marry the imaging, the PET imaging notably, the MRI, but also the motor and cognitive assessment of these non-human primates, and all of this under confinement, biological confinement that were required. And so we built Myrcène as such to produce from in-cellular models up to preclinical animal models, rodent and non-human primate, everything necessary to try to develop and test and validate cell-based, gene-based, and obviously drug-based therapies. So this is Myrcène. It's not a molecular imaging research center only. It's a real translational preclinical center. All this is combined also with strong collaboration, long-lasting collaboration to more than 30 years with clinicians, colleagues that were trained in Myrcène. They were doing their PhD thesis with us, and then later on they moved to a neurosurgical or a neurological department. They were head of those departments and continue to collaborate with us.
And what is impressive is that here you had to get very specific capacities, very huge, large ones to welcome, for example, primates. So can you tell us about the capacities, facilities?
Oh, the facility is obviously a bit large, I would say, for France. It has been constructed as in a way that we could run many projects in a row, notably in non-human primates. So we have 400 primates, for example, in different conditions. So we can run up to 10 projects simultaneously in non-human primates. We also include in those capacity the possibility to run projects in rodents, of course, mouse and rats and all this stuff.
And the link with clinicians is Neuratrice?
Yeah.
You run Neuratrice?
Yeah, I run Neuratrice. That's another aspect which is interesting for industrial partner, of course, because Neuratrice is an infrastructure, national infrastructure of translational neuroscience. So dedicated to neurodegenerative disorders. And this regroup four Parisian hospitals and 3 research centers in the Paris area. So we are all together in the same geographic region, so capable of interacting very smoothly. And this Neuratrice infrastructure, which is coordinated by Myrcène and by myself.
You coordinate yourself?
And well, I try to do so. But so we have this opportunity to really run together translational projects from in cellular to clinical application, phase 1 trials.
Phase 1 trials. And you have some interactions with industry, of course, especially Sanofi, I think, but not
Well,
other
it has
partners?
been Sanofi
It has
for a
been,
while, and now it's more Servier,
Servier. Okay,
which is hosted in Mersenne for different reasons. And actually, if I would comment what industry bring us is a kind of collaboration, trying to develop new tools that could be useful for Mersenne, for example, but also for the industrial partner. And for example, if I take the example of Servier, what we do with Servier is we do two things. We collaborate on building new imaging tools to serve their purposes, and we run their own project in Mersenne using these tools that we have been developing together. So they have full confidence in the tool that we are going to use because they co-develop it. And for us, it's interesting because we can publish this out of the
It's not only service, it's a cooperation.
So it's a collaboration in the first step, and then it becomes for Servier in application, of course. So that's one example of what we can do with industrial partners, of course.
Roger Legrand, I said you're head of a division, or you can take another mic as you want. You're head of a division of human virology at CA, and especially you run eDMIT platform, preclinical research on infectious diseases. Can you tell us more about this huge platform?
Well, this platform is quite similar to Mersenne, but applied to the field of infectious diseases and immunology. And our aim indeed is to increase the expertise in the preclinical domain of infectious diseases, and then to propose this expertise as a support for programs that are bringing through academic programs or industrial partnerships. And in terms of the size of the activities, so we have recently become the largest preclinical infrastructure in Europe for performing such studies.
So it means that you're very well connected to industry, and even we talked about the entrepreneurial spirit. You are connected to this spirit. You have infrastructure to help you, administrative ones. You can run like a.
Well, I think that to be in the translational activity and to be connected to the industry, what we need first, and that was already emphasized by the previous speakers, is we need the expertise and to be, and this is what the people look for. So they look the experts, and not only the facilities we have. So the facilities are very important, but if you don't have the people to run the facility and to bring something new to the company, I think this is very important. Second, we have to organize ourselves to be in a position to accept the constraints of the industrial partners in their drug development. I mean, in terms of quality assurance, what we provide, in terms of timelines. That's certainly a main issue in many academic private partnerships. And we need also to find a way to position ourselves in the whole picture of the ecosystem. When I'm saying that, it's very important, at least for my view in these positions, to not be in competition with the industrial partners when we are doing this partnership. We need to bring something that they don't have, and they need to be asking us something that they are looking for. So I think that's, and this is not only an offer or a talk like today, it's also long-term work we have to do together.
It's a matter of trust.
It's much as trust. I think there are several things. I like the way that we are working with Sanofi Pasteur, for instance, at the moment, or GSK. So we have, as Philippe said, collaborative programs. So what we do with collaborative, we create the tools, we increase the expertise together in the mid-long-term programs, 2 years, 3 years, 5 years. And then we can enrich the knowledge, and then they will use these tools and this knowledge for their own purpose, for the development of their own drugs as a service. And this is, I think, a very good deal between us, because we are indeed producing knowledge, which is our prime mission, but we are also producing a support for the companies. We can also provide this support for academics. And then I think what is also very important is that by doing this, we create a proximity between the scientists of the industrial partners and ourselves. Because it's not just a command and from our side providing a quotation of what we can do or whatever. At some point, when we do some kind of collaboration, we have common ideas. We share our expertise, we share our ideas, and we can elaborate a strategy for the future. And I think this term of strategy, this is more or less what was lacking during pandemic situation here in France.
By the way, as you talk about the pandemics and the COVID, you have another project which is Gaston Ramon Institute.
Yeah.
And I think it would be interesting to talk about it with you, Jean, because I think you have your idea about that. But it is about vaccine developments. Can you tell us just a few words about Gaston Ramon?
Thank you very much for this question, because Gaston Ramon is still a project at the moment.
Right.
But actually, this is the idea, is to really try to stick to what I was saying recently, what we need or what we learned from the pandemic, that we have an ecosystem, which could be excellent ecosystem, but we have some gaps. These gaps are more or less identified right now. For instance, for bioproduction, we don't have enough bioproduction capacities in Europe. That's for once. The other thing we lack, I think, is the organization of this ecosystem as a pipeline. So we need to make this ecosystem agile. And how we can do this how we can make this economic agile is by connecting the bricks. So people who are doing upstream research need to know what's happened downstream in the preclinical world, in the clinical world, in the bioproduction, so that they can anticipate what the constraints for the bioproduction where they design the antigen, for instance. This is very well known in the industrial area. But these people need to be connected, so they need to be all together. And we need a kind of common process management to transfer the activity from one part to another very agilely in days or weeks and not in months or years. And this is very important. And this is what Gaston Ramon will try to perform if we succeed to get this project, is to really try to organize, connect, and then offer this organization as a support for the companies. The other objective of the Gaston Ramon is to try to respond to the strategic needs of France in developing vaccines, for instance, in the fields where the marketing is not going. So molecules or instruments or vaccines that are not covered by the market.
Tuberculosis for adults, for example?
Well, tuberculosis could be one example, because tuberculosis is probably a very difficult vaccine to perform. So it's not easy for a company, even a large company, to invest a lot of money without a visibility in the returns of this strategy. So I think here the public-private partnership takes all the essence of what we can do. But there are other fields, for instance, anticipating the emergencies in terms of infections. So you need to risk or bet on a class of pathogens. So it's not always easy to produce a vaccine that will stay in the garage and will not be able to sell it. But we need this, and we need to demonstrate that this vaccine can be produced at least at a small scale, and we need to demonstrate how we will organize a scale-up if in case of needed. And the other field of illustration is that, for instance, the bioterrorism threats, we need some vaccines for bioterrorism threats. And there is, I think it's not the mission of the large company to really work on this unless they have large companies.
Bioterrorism. Mm-hmm.
But we can prepare that in an area, in an institute like Gaston Ramon, in order to be prepared and then transfer to the companies when the threat is coming.
Okay, we'll go back to this institute project, namely with Jean Lang, who's very much involved in vaccine development. But Nicolas Tournier, first, you are imaging pharmacologist here, and you developed your activity during the video, which is very interesting. We go back to the spirit, I'd say, of this roundtable, which is translational medicine, but also phase zero. And you are very connected to this idea of phase zero. Can you tell us what is phase zero for you, and how do you help to go quicker, faster? This is a motto that we have since the beginning of the day, and to be quicker on patients. Can you tell us?
Yeah, sure. Thank you. Yeah, I come from the PK, the DMPK, and I learned a lot from the PK before coming to the imaging community. And the first people I met in the CA were radiochemists. There's a long history of radiochemistry in the CEA. And they have fluorine chemistry, but also carbon chemistry. And most of the drugs can be radiolabeled. So imaging in most situations is dedicated to one target. Can be a virus, an infected cell, can be a neuron, for instance. But I think the drug can be the focus of the imaging if you radiolabel it, and you can gain very early clinical data. And we are lucky to work in a center where imaging can be performed in animals and humans. And the phase 0 design allows for quick transfer because it's microdoses, so the regulation is very open and lighter than conventional phase 1. So I think phase 0, when possible, provides a shortcut to go from the very early and sometimes in vitro data
because
to clinical.
Microdose on the whole body and not only in a blood sample. You can have a very early view of its action. That's it.
You don't really have a view of this of the action because the dose you inject do not have effect. Yeah. It's mainly distribution to tissues, but when used for PET imaging, you have distribution not only in the blood but also in target tissue, vulnerable tissues.
So this is what you're looking for.
Exactly.
And this procedure, this platform service, do you already have some industrialists involved?
Not so much in France, but I think there's a community that is building in the world on the concept of phase 0. Many researchers from academic and industry talk together because it's not well accepted yet in most in the normal and conventional development of new chemical entities.
Why?
I think it's only habits, and it's not always possible. It's very important that it is possible in some specific situation where radiolabeling is possible. But when it's not possible, you have to go the conventional way. But when it's possible, in my opinion, it's a unique opportunity to select one compound, go quickly to go-no-go decision, and earn time.
Okay, so we'll go back to you too, and we'll ask if you're interested in this phase 0 approach in a few minutes. But Vincent Lebon, to finish talking about the platforms and CA. So you're a kind of synthesis of various platforms we talked about. IDMIT, Mersenne, here present, Neurospin, we talked about, through Passerelle. What is Passerelle exactly? What do you do exactly? I reminded you, Professor of Medicine at Paris-Saclay, Head of Department CA, and you co-lead this Passerelle platform. What is it?
Thank you, Fabrice. So I think clearly CEA offers unique platforms for biomedical research, as you said, Mersenne, IDMIT, Neurospin, SHFG, outstanding platforms and devices for biomedical research. Our challenge is really to bring those tools to patients and hospitals, because we are far from patients and hospitals. So I think this really is the main objective of the Passerelle project, is to connect, to transfer, to translate our technological developments to patients and medicine. And for this, the idea is to create an ecosystem, and I think this is a direct answer to the comment by Ramzi Temani from Janssen, just the previous roundtable, who was requesting the building of ecosystems, but not only academic ecosystems including industrial partners and hospitals. And that's exactly what we want to do through the Passerelle project, is to connect the three partners that are academic platforms, industrial partners, and hospitals. And that's the main purpose of the Passerelle project. which is supported
So
by...
you recruit patients and you are in clinical issues, that's it?
We actually federate networks of hospitals in the Paris-Saclay region. We connect them with academic labs and platforms. And we also work on developing the interactions and the collaborations with the industrial partners. Because any innovation and development will never reach or spread to patients without the input and the contribution of industrial. So within the large project, which is Passerelle, which is a project which is strongly supported by CEA, of course, but also by the university and other national research organizations, we have a specific program focusing on medical imaging and on drug development. And this program really starts with the conviction that our advanced medical imaging techniques provide invaluable information to accelerate and de-risk drug development. So we've been starting working on this program with industrial partners too. And the idea is really to set up a new paradigm for these collaborations between our academic platforms, imaging platforms, as we said, Mersenne, IDMIT, Neurospin, SHFG on one hand, and with drug companies on the other hand, to open our platform to accelerate those developments. So we'll work on developing a new governance paradigm, governance of the platforms, including or integrating industrial partners, developing also a clear offer of imaging services for the industrial partners that can be accessed by or throughout a single point of contact, which is something really important. We are 4 different platforms, and I think unique access for industrials is something really crucial, and easy access. And finally, we're also working on a dedicated high-level training for the industrial partners who sometimes have difficulties to really understand the potential, but also the limitations of our advanced technologies.
Okay.
It's really a brief summary about Passerelle.
Training for you, Jean Lang. Maybe you will be trained. You're Associate Vice President, Global R&D within the New Vaccines Project Department of Sanofi Pasteur. And you must be listening to this conference with a great ear, because you want a virtuous circle and to put together academics, agencies, and industrials.
Yeah, let me first acknowledge the organizer, because it's a great pleasure as a private sector scientist. I'm a scientist, I'm an MD too, to meet the colleague from the public sector. I've been trained to be a public health official. Now I'm in the private sector, but we are scientists driven by science, quality, and timeline and investment, of course, because we're in the private sector. And why do we collaborate with CEA, and Roger Legrand in particular? It's not because he's French and I'm French, it's because it's one of the best teams in the world. And we, as a global company, as an R&D company, we need to pick up the best talent, the best brain, and they need to be a joint value proposal, project proposal. We are not doing that for the sake of investing in CEA or CEA to invest in Sanofi, but because together we can bring value, and value is really the guide, the key performance indicator we want to achieve together. And looking at Roger's platform, it's unique, it's competent, it's quality-based, it's timely, it's efficient. What we are looking for in translational science and vaccine, which are prodrugs, elicit immunological response, are driven by translational medicine, because, as you probably know, there are plenty of vaccines very, very efficacious for mice, but the problem is that they are not working in humans. And by the way, that was, you know, the fact that mRNA vaccine wasn't working was not a slam dunk at the beginning. There was a lot of iteration, but when we saw the translational monkey data and NHP data, we were convinced that it was somewhere provided there will be translatability. So what are we looking at this model, working with CEA in particular, because we are looking for several parameters. Number one is to fast to fail or candidate. So we don't want to invest into a vaccine that will fail later on in phase 3, as occurred, for example, in our Clostridium difficile vaccines.
Yes, unfortunately.
So that will really help us. And number two, it's fast to win. So we are reassured, because we want to generate in the vaccine area biomarker. We want to generate efficacy, safety, and immunogenicity biomarker. We want to down-select candidate, because we, as you know, it's a combination of adjuvant for some time and antigen. And last but not least, we want to decrease our granularity to understand the interactions between age, between pregnancy, for example, which can be achieved in the NHP model, in this real human model. And that all that, the translational science, the phase 0, will help us to de-risk. And we have one very concrete example I just alluded to, it's mRNA. Everyone is investing, and we are heavily Sanofi investing in mRNA. We have invested 400 million euros, 400 people in this mRNA. And we are working
Right at the beginning, like Pfizer or Moderna, for example?
Not right at the beginning. We're investing in a platform in T-Bio and in the COVID-19 vaccine antigen. But then we realized that BioNTech, because that's Pfizer is a BioNTech vaccine, and Moderna were far advanced, and we need to have a differentiating offer that what we are bringing to the table with the subunit adjuvanted vaccines.
Did you think that the problem that happened with your vaccine at the beginning was connected to what you talked about, the environment, the ecosystem, sometimes difficult to work together and to be really connected?
In fact, we were connected to the ecosystem, and as you know, when the third partner, the third pillar of the vaccine, besides Sanofi vaccine and GSK, was BARDA, the US BARDA. And it was a good line to criticize BARDA because the US paid, but this is remarkable. And in fact, Ursula von der Leyen realized that we lacked that instrument in Europe and maybe in France. And what Roger envisioned, and we envisioned together.
You want to make a European BARDA?
Not me. The European Commission has already set up the principle against
cross-border. But you're a great supporter of that.
Yeah, it's called ERA, Health European Preparedness and Response Authority, like BARDA. The Biomedical Advanced R&D Authority. So we have that, we have the principle. It's a new directorate in Brussels, so it's far advanced. Now we have to fuel it by public-private partnership model. And what we need to build now, the next step for France, I'm calling as a French scientist, we need to build this capacity with NRS, maladies infectieuses émergentes, and IASDA, Pana. And Institut Gaston Ramon could be a part of that, focus at some technology with VRI from INSERM. But we need to think broader than that and build the biocluster, the infectious disease biocluster between Lyon and Paris. Where together we build a critical mass and we think beyond our own silo. I'm a private sector scientist, I'm a public sector scientist, because what joins us together is to achieve competitiveness. And for the first time in ERA and IHI, the new public-private partnership in Europe, you have the word, make Europe competitive, together. And that's the shared visions I would like to develop, and CR and Sanofi will be an integrated part of that.
Okay, that's pretty clear. And we'll go back to this idea because I think you must be very interested in it, of course, Roger Legrand. Magnus von Tess, you have your point too about translational medicine and especially reverse translation paradigm. And you have, I think, million patients around the world in clinical trials. And you want to share this view.
Yeah, I can echo what Nicolas Tournier said a while ago. I mean, a very good model for human disease is human disease, and yeah, and obviously it's not the only tool, but it's very important for us now, and we can look at what is happening in Immuno Oncology as an example. So around the world, as you said, there are around 1 million patients in a lot of different clinical trials involving immunomodulation. And this is, of course, quite unique in the history of health. And we are actually actively modulating the immune system and measuring what is happening. So we will not only learn about cancer and the particular patients, we will learn in general around the immune system. So this is what we hope. So a lot of fundamental knowledge around the immune system, we hope, will evolve from this. And I think an important aspect here is also and I think some low-hanging fruits, both for academia and for industry, for all partners in this ecosystem, are longitudinal sampling. So when we look at disease and health, it's a dynamical thing. It changes over time, and often we only have snapshots. We have one snapshot. Maybe when the patient came into the clinic, we go fairly deep. We have a tumor biopsy. We blood draws, everything we can do at that moment. but we often lack the longitudinal following. And it's a dynamical change. And I think that just doing some dynamical modeling and really following the time course, there is low-hanging fruits to pick. We might find new biomarkers that can lead us also to new targets and also advance, I mean, the understanding of immune systems and health in general. So I'm quite excited to be alive at this time point.
Great. It's optimistic and positive. What about the platforms you heard about and the help that could be for you?
This is key, of course. I mean, if we even so, I work for Roche, and we have a very big internal ecosystem. But even in our areas of high value, we represent a little part of what is done in the world. So we need to work as an ecosystem, engage with partners, and there are multiple aspects here. Of course, there are platforms that will help us to get data and the quality here is key. I mean, crap in, crap out. This is always what happens when you do data analysis. So we need really good data, but we need to set up what kind of data we actually collect. This is, of course, very important. So we need the field's expert, both maybe internal experts from I mean, biotech and health industry, but also academics, that we together map out what kind of measurements we want to do. And then we need those measurements. And then we need to work together also in this public-private interface when we do the analysis. We need field expertise as well as computational expertise. I would say that the targets for us has not changed. We want to understand biology, we want to help patients, but the tools we have are advancing rapidly, so that is what's changing.
Thank you. A few questions before the end of the round table, because we had a great view of all what you're doing, but there was this question of biocluster in infectiology. What do you think of it, Roger Legrand?
Well, I think there are, at the moment, different initiatives and inspirations for trying to prepare the future and how to face this competitiveness, of course, but not only for the market, also against the pathogens. And that's what happened with the pandemics. We were not prepared enough, so we need to be fast because we want to control fast also the pathogen. And I think these different initiatives are very complementary each other, actually, because we need a driving force for the companies, that's for sure, and for the private sector. There will no vaccine delivered to the world or for mass vaccination without the private company. This is not, we cannot imagine that. On the other hand, we need also driving force for the strategy of France or the Europe, and we need to connect these. And I think all these initiatives are responding to the same purposes. This is what we can drive by the companies, what can be bring by the academics, and how we can connect each other. So he was talking about bioclusters. We were talking about an institution, Ramon Institute, but these are not competitive entities. They are really focusing on complementing targets.
Complementary. You agree with that. A few questions coming from you, connected in France and from Boston. I think you've already started answering to that question, but do you have any international connections already in phase 0? I think it's already the case.
We have academic connections, international in Europe, inside Europe, with Vienna in particular. We have a phase 0 design study with patients that are difficult to include, and so we do B-centric using exactly the same methodology in both centers. It goes twice faster. And we also worked with an academic lab in the US, wanted to validate in vitro models from human data. They were studying liver transporters, and so they used our imaging data as a gold standard to validate the in vitro model.
Okay, there is this interesting question which is very open. What are you missing that we can provide? So I think it must be from Boston somewhere. I don't know if from the US, or do you need something? Yes, Philippe Entraille.
Yeah, maybe I can answer that question, or at least give what I think we are missing. We are missing actually good ideas to test for with partners that are interested in these ideas. We need also new targets, maybe. We are mostly interested in developing biomarkers, mostly early stage biomarker. And of course, we know we are looking at the pathologies, and in Mersenne we are more interested in neurodegenerative disease. And doing so, I think it's very important for us to see what are the kind of target that are interesting the industrial partners to be able to develop those biomarkers either by PET or by MRI actually, and test if they are really predictive and early, I would say. Looking at early neurodegeneration is a key issue when you want to do neuroprotection, for example.
Okay, thank you. We have 3-4 minutes left. Do you have a one-stop shop for contracting? Vincent Lebon, I think you have an answer.
Yeah, I think it refers to the idea of a single point of contact for contracting with CEA imaging platforms.
Okay.
This is clearly a priority for us, and we are actually working on a quality certification of the one-stop shop process for our platforms. So yes, very soon, coming soon.
Coming soon, okay. It's
certified.
Okay. The last question we have, there are other questions. Thank you very much for participating. Do you practice open data? So it's for all of you, and especially in industry. Magnus.
So the answer is, I think that we have seen examples here of when we work together in a pre-competitive space, and all big companies engage in this. So basically, we build value together. Often this results in a lot of open publications, but then every partner brings with them value that they can develop internally. So this has to be done it's a collaboration, but everyone benefits. It has to be regulated, but it's absolutely doable.
Okay.
Yeah, usually in the large European public-private partnerships or American public-private partnership, we define from the beginning legally who is on the data, who the data will be sensitive, because some need to be protected, but the basis is that you bring the data, you bring the value, you own the data, and you can share largely.
Okay, and we have a last question. Just time is nearly over. Do you have a strategy in cancer, rare diseases, and autoimmune diseases? It's a very open, wide question. Somebody wants to answer quickly about that? Magnus von Tess.
Obviously, the answer, I think, for all big pharma companies is yes, we do.
I agree.
We think carefully about this in collaboration with a lot of external partners. And it is about what kind of measurements we make, and how we sample, how we create data, and how we analyze that data. And we always have clear questions in mind. So the answer is yes.
Yeah, for rare disease and autoimmune disease, I could even include neglected topical disease. This is a share value project we need to achieve through mainly public-private partnership and push and pull incentive that could de-risk the collaboration.
Thank you very much for your participation. Philippe Entrailles, Roger Legrand, Nicolas Tournier, Vincent Lebon, Jean Lang et Magnus von Tess. Merci à vous. Vous pouvez regagner vos places parce qu'on va directement enchaîner avec la fin de cette conférence. Merci, vous pouvez les applaudir.